Good morning and welcome everyone. Some housekeeping to start. Can I remind you to please type your questions into the chat and we'll have time to address them during the webinar. Our Q&A moderator today is Dr. Will McCants from Barwon Water. In the Barwon system, the Upper Barwon is one of the few places where you can safely drink from the stream. The Barwon, Yarrawee, Lee and Moorabool rivers suffer from multiple pollution sources. Why should fish and other aquatic biota consume this chemical cocktail that will jeopardise human health?
We're just going to go to a short video. We'll send it around afterwards if there's any glitches that you experience on your end as well.
We're walking beside Lake Elizabeth, high in the Otways. Lake Elizabeth was formed across the East Barwon River in 1952 by a natural landslip in a year of extreme rainfall. It's a beautiful, biodiverse bushland refuge for numerous platypus and native fish.
Welcome to our Nature Positive webinar series. The Nature Positive initiative is a United Nations-backed global society goal to halt and reverse nature loss by 2030. It recognises that nature and climate crises are deeply linked. Hi, I'm Locke Gordon, Acting President of Friends of the Barwon. Friends of the Barwon was born out of Landcare and is a catchment-wide community group working to protect and restore the waterways of the Barwon system. Friends of the Barwon works on the lands, waters and seas of the Wadawurrung people of the Kulin Nation and the Gulajan and Gudubanud people of the Maar Nation, and we acknowledge them as the traditional owners. In 2014, I set off with a small group of land carers in the Otway Ranges, close to the source of the East Branch Barwon River, upstream from Lake Elizabeth, to walk, raft and kayak to the sea at Barwon Heads. Each year we travelled for 3 to 4 days, and in 2017 we reached the estuary at Barwon Heads. Along the way, we spent time with Wadawurrung traditional owners who introduced us to some of the remnant native food sources. In 2018, I wrote the following passage in our book, Walking the Barwon River from the Source to the Sea. Our journey revealed places of stunning natural beauty, as well as barrenness, pollution and degradation. We could only imagine what the river must have looked like and represented to the Indigenous inhabitants only a couple of generations ago. It's to our collective shame that we've allowed such destruction to occur, and it's incumbent on all of us to redress the damage we've done and are continuing to do. Friends of the Barwon adopted the Nature Positive theme for this webinar series to explore how we can halt this damage and reconstruct the Barwon waterways ecosystems to return them to a condition of productive, self-regulating biodiversity. I thank Barwon Water for help with facilitating this webinar, and I dedicate this webinar series to the memory of Dr. Trevor Hodson, late President of Friends of the Barwon. Our first 2 webinars will dispute the old theory that the solution to pollution is dilution. Dr. Benjamin Long of Federation University has a special interest in the fate of pharmaceuticals in the murky waters of the Yarrawee Lee. Dr. Long.
Okay, hi everyone. So I'll just try and share my slides with you all. Just confirming, are everyone able to see this correctly?
Yeah, thanks, Ben.
Yeah, thanks, Will. Yeah, thanks for the introduction, Locke. And I have a special interest in the Yarrawee and Barwon River system. It's a place that we've been studying, and also I've grown up around my entire life as well. So I'm going to talk to you today about some studies that we've done along this entire system, a little bit about the concentrations of the pharmaceutical compounds that were found there, plus a little bit about one of the potential fates that they have. So pharmaceuticals are what we would class an emerging contaminant in the environment. Now, this isn't necessarily because we haven't known about them for a long time, because we have. We've known about them for years. But what we don't know is a huge amount about their full distribution, where they end up, and then what their full effects are. Wastewater treatment plants tend to be the big vector for these contaminants into the environment. Now, I say vector because the source is actually you and I. So when we take pharmaceuticals, our body processes them, and then we put them into the toilet. Gets flushed down through the sewerage and to the wastewater treatment plants. Now, they're not traditionally designed to get rid of these small amounts of pharmaceuticals, and so they do get passed through and discharged into the environment. Some wastewater treatment plants are good at taking out some of these pharmaceuticals, but not all, and so they do end up out there. Now, the levels that we're talking about are what we would call parts per trillion levels in some cases. So really, really small amounts, basically. I'm not going to focus a huge amount on the actual amounts out there, but we're talking in terms of, masses. We're talking nanograms per litre or nanograms per kilogram. So very small amounts. But it's these amounts that have still been shown to concentrate in freshwater as well as marine organisms. And these are well below what we would define as a lethal level for any of these drugs. But it's not below what we would define as a sublethal effect. And this is where the potential issues for these pharmaceuticals really come out. I've got a picture of a marbled crayfish on the slide. Now, this isn't an Australian native. This is actually a European crayfish, but there have been studies done on this crayfish showing that these environmentally relevant levels actually affect the behavior of these crays. So they're slower and their hiding behavior gets changed. And so this changes how they feed and they are preyed upon. So it can affect ecosystems from that perspective. And so rather than it's a lethal effect, we're seeing sublethal changes to the ecosystem here. So this brings me to why the Yarrawee and Barwon River system is of interest. So in addition to what I said about before and what Locke has said about it being a fantastic river system, it's also a really unique opportunity to study because we've got 180 kilometers of river system, but it only has one point source of pharmaceuticals or point vector, which is a wastewater treatment plant up near Ballarat. And we wanted to look at this and trace just 3 pharmaceuticals, 3 commonly prescribed pharmaceuticals, and see how they behaved all the way down this river system. The 3 that we looked at were carbamazepine, which is an anti-epileptic drug, tramadol, which is an opioid pain relief, and venlafaxine, which is a antidepressant. And all of these are within the top 50 of the most prescribed drugs in the Australian Pharmaceutical Benefit Scheme as well. So they're widely prescribed all over Australia. And so what we looked at was, I'll get a laser pointer out for this, is we looked at basically the whole Yarrawee Lee river system to where it joins into the Barwon and then out through to the ocean. And each of those dots are where we took samples from. Now, there's a couple of sites of, I guess, special note, we basically spread them out evenly as possible, taking into account a bit of access requirements to the river. But we've got one site up here right at the top, which is before, so it's upstream of a wastewater treatment plant. Y2 is actually a wastewater treatment plant, so the water that is being discharged into the river. And then we also have some measurements from the Upper Barwon River, as well as the Moorabool River as well, which in theory, neither of these places should have, neither of those three places, the one upstream or the Upper Barwon or the Moorabool, should have pharmaceuticals in them. And the good news is they really don't. Neither the Moorabool or the Upper Barwon have any pharmaceutical contamination that's worth noting. There's nothing that was below our detection limits. What we did find, though, of course, was a reasonable amount of these pharmaceuticals close to the wastewater treatment plant. But the further we get away from the treatment plant, the less pharmaceuticals we found in the water. And so, bringing that back to that is dilution, the solution to the pollution, as Locke said earlier. We do see a lessening of the amount of the pharmaceuticals the further we get down the line. We didn't see any real correlation between the season that we sampled in, because we sampled across numerous seasons, or any of the water quality parameters that we measured either. So we didn't really see any huge changes there. But what is interesting is we did see, based on the amounts of pharmaceuticals in the water, we could separate, using some statistics, the river into three regions, which is the upper reach, the middle reach, and the lower reach. The upper region was like the true Yarrawee-Lee system. The middle reach was between, it's the Barwon, between Inverleigh and Finesford, and the lower reaches was Geelong through to Barwon Heads. And I think those also make really nice sense in terms of regions of the river as well. So we wanted to look at these regions in terms of a risk-based approach. So whilst we've got some numbers here, what is the actual risk? And so this is a part that I am going to focus a little bit on the numbers rather than just trends. So we used a process called a risk quotient to look at this. And this is a ratio between how much is actually in the water, so what we measured, and then how much, well, what's the line where we would expect no effect on the wildlife in the water. So it's a predicted no effect concentration is what we call it. And when you take that ratio, basically, if you get a value larger than 1, or a value over this line here, that's a higher risk scenario. Anything below that line, you've got an attenuated risk. And anything below this line here, we would say it's a limited risk. So we don't expect a risk. And so it's these concentrations that above 1 is where we're seeing those non-lethal effects. We're seeing altered gene expression, perhaps a change in growth, or behavior in aquatic organisms. And these are model organisms. These aren't Australian natives necessarily. Before I get to that I've just put up, I will show that carbamazepine in all reaches of the river was found to be a high risk scenario with little attenuation down the river. Conversely, tramadol, the opioid, was what we would call a moderate risk in those upper reaches. But as we moved further down the river, we went down to a what's called an endurable risk to the system. And venlafaxine was, I think, in terms of risk profile, was the more interesting because it is high risk at the upper reaches, but then we get down to a more of a moderate risk as we further get down the river. So this, I guess, dilution, if that is the only effect, is we are seeing a change down the river. None of this takes into account synergistic or antagonistic effects, which means that we're not assessing how these behaviors are mixture, because we've got all three of those there. It's not just one component. That's a very understudied area though. Some things are worse when they're mixed together. Some things are better when they're mixed together. So for example, caffeine tends to be have a protective effect when you've got other pharmaceuticals that may have those sublethal interactions. So at this point, we know the pharmaceuticals are present. There is some dilution effect there for sure. But I think if it was dilution only, we would see these sort of large decreases in the concentrations as we hit those major confluences. So at Inverleigh and at Moorabool, but we don't really see that. It's not clear. And so we wanted to explore some of the other potential fates. So some of the other potential fates of these pharmaceuticals are that they just get broken down in the water by water and sunlight. They get uptaken by sediment or plants. And plants is what we wanted to look at. And I think it'll become obvious why we went down that thinking route a little bit later. So again, studying the same system, many of the sites are shared here. But the thing that I want to focus on is that we looked at these two species here. Whilst we collected a range, these are the two most interesting and prevalent at each site. So we've got Phragmites australis and Villicinaria australis. So the Phragmites australis is this common reed species. Might be hard to see in this photo. This is taken at Finesford, but it's basically everywhere. Sometimes it grows with Typha, which is another one that we sampled, but it grows everywhere. It actually grows everywhere around the world, despite the species name of australis. It's a cosmopolitan reed. The water ribbons, however, these are a submerged plant. You can see them laying down on the top of the water there. That's actually a little bit more special to our location. It's endemic to southeastern Australia and was actually used as a food source traditionally as well. Now, what we saw with these plants is that, oh, it's refusing to change. There we go. They were uptaking carbamazepine. It's the only example I'm going to show for this section. And again, it was much higher at closer to the wastewater treatment plant and less further away from the treatment plant, which I think we can probably all expect. There's less in the water, so there's less to go into the plant. But they were being uptaken and by quite a bit more. So we were seeing levels of, in the less than 1,000 nanograms per litre in the water, but we're seeing up to 30,000 nanograms per kilogram in the plant. So they are uptaking it quite a bit. And we found we term this a bioconcentration, and we can look at how well they actually do that. And we see a 50 times increase in some plants, but up to over 300 times increase in other plants. And we actually saw this effect increase the further we went down the river, which is, from I think a plant physiology perspective, is quite interesting. And it's not something that we have a definitive answer for, because it could be based on actually how the plant behaves when exposed to these pharmaceuticals. But also, I think our most probable explanation for this increase in bioconcentration as we get down the river is that these plants are exposed for their entire life cycle. So from the point that they come out of a rhizome, up, sprout up, and then die off, they are consistently exposed to low levels. And so they're constantly uptaking and constantly concentrating these drugs. So I think now's a good chance to say, why did we go down this route? Why did we specifically want to look at the plants and not necessarily those other methods of fate? And it's because if it's in the plants, it gives us a potential point of which these drugs can enter other food webs. So could the things that live in those plants or feed on those plants be getting extra doses of these drugs by drinking the water plus eating the plants? And so it's moving into the food, further up the food web. It can also be a point of where we transfer into a new food web. So moving out of an aquatic environment and into a terrestrial environment where things are living on the land. The other reason why we wanted to look into this is for a bioremediation factor. So could we use these plants to actually take out the pharmaceuticals? So could we design wetlands and things that we could actually remove the pharmaceuticals from wastewater streams, for example? And as a bit of a summary, so coming to the end of what I wanted to talk about today, we found pharmaceuticals in this system with some decrease in concentration, but it is, especially in that upper region, more of a high risk scenario. And we also found carbamazepine, and didn't actually show you venlafaxine, but forgot to take this out of the slide deck, were found to be bioconcentrating in the Phragmites and Villicinaria species. And brings me to my last slide, is a bit of a where to from here slide. Because we have found this bioconcentration, we are currently looking into whether we can use, say, the Phragmites australis for the bioremediation of pharmaceuticals. So building some floating wetlands for that purpose. Plus, we're looking into the trophic transfer of pharmaceuticals. So are these pharmaceuticals actually getting out into the food web, or are they staying where they are? So that's to help us assess how much of a risk that these pharmaceuticals are. And so both of these are actually in collaboration with Barwon Water. And so we're hoping to have some results with them relatively soon. And with that, I need to thank the people who actually did the work. So Ahmad, who's nicely photoshopped into this photo here, did the first study, and Sam, who was responsible for that second part of the study. And of course, sponsors, Barwon Water and Central Highlands Water, and of course the Wadawurrung people, who were the traditional owners of the lands of which we did our studies on. That's all I've got for you. Thank you very much for having me.
Dr. Aaron Schultz of Deakin University is developing baseline case study data for metals, agrochemicals, pharmaceuticals and personal care products and microplastics in Lake Colac. Lake Colac is connected to the Barwon River via the Lock Calvert drainage scheme, which is only activated when the lake reaches a high water trigger mark. Dr. Schultz.
Thank you, Lachlan. Thank you, Lachlan, Will, and friends of the Barwon for the invitation to be here today and present. So I'll just get my slides shared as well. So hopefully you can see them now. and hear me okay?
Sure can. Thanks, Aaron.
Perfect. So yeah, thank you again for the invitation to present today. So I'll be touching on a collaborative project that's currently underway between Ben, who you just heard from at Fed University, as well as Barwon Water, where we've got two amazing PhD students working on this project. At Deakin in my lab, we have Nishat Ermi. She's doing a lot of the work that I'll be presenting today, and as well sort of finishing off some work that we will hopefully present in the future in this space. And then over at Federation University in Ben's lab is Sam Harridge. And so again, that's some of the data that Ben was just presenting previously, and she's working on this project out at Lake Colac as well. And the overall sort of theme of this project is understanding and knowing that healthy catchments and healthy ecosystems are important. And so what we're trying to do is understand the different contaminant levels that we have in these regional waterways. And then future thinking is then developing strategies for success to try and, develop strategies for bioremediation, as well as minimizing the release of these contaminants into these important ecosystems moving forward. And so Lake Colac is the site that I'll be talking about today. So Lachlan's already done an amazing acknowledgement of country, so I thought I'd do something a bit differently today. So I am presenting today from Wurundjeri country. However, I became aware of a wonderful sort of snippet from a report that was produced a couple of years ago by Dr. Rebecca Nelson, Professor Lee Godden, and Dr. Bruce Lindsay, that was part of the Natural Cultural Flows Research Project. And so I'll just read a snippet of that, just highlighting the significance of our freshwater catchments to First Nations people. And so what they state is that for First people, water is a sacred source of life. The natural flow of water sustains aquatic ecosystems that are central to their spirituality, their social and cultural economy and well-being. The rivers are the veins of the country, carrying water to sustain all parts of the sacred landscape. So again, just from that quote, you can see just how significant freshwater environments have been to the First Nations people, but more importantly, they're again a really important environment that we again need to protect and ensure we protect for future years and into the future. So at Deakin, my research group, our philosophy is using a One Health approach to tackle pollution. And so this is extremely multidisciplinary, but what this does is it takes in sort of the belief with the World Health Organization's definition that One Health is an integrated and unifying approach that aims to sustainably balance and optimize the health of people, animals and ecosystems. And we know that for us to have a healthy society, healthy population and healthy individuals, we need to make sure that we're protecting our environment so that we have healthy ecosystems, as well as protecting the animals so that they're healthy living in those environments as well. And so the research that we conduct at Deakin within my group is bringing all of this together, where we're able to use human and animal cell lines in culture to look at pollution and toxicity risks of different contaminants within the lab. This helps us predict any risks that could be posed to human health as well as environmental health in the future. We also have projects measuring contaminant levels out in freshwater and marine environments. It forms part of this healthy environment umbrella. And then we also have projects where we have model organisms in my lab that provide a really valuable tool for looking at the potential risk of different contaminants to aquatic organisms. And the main model we use are what's called zebrafish embryos. So zebrafish are a freshwater teleost native to India, but they are globally used in research as a really important scientific animal model. They are actually sustainable, but more importantly, up until 7 days post-fertilization, they are actually classified as non-living. And so they're an ethical model for using and studying toxicity risk as well. This is an image that one of my PhD students captured that shows fluorescent microplastic uptake. So again, they're really robust at being able to track pollutant levels and concentrations in different animals as well. For today, though, I will be just focusing on this sort of part of the research that's conducted, where I'll be talking about the work that we're doing out at Lake Colac. So Ben's touched on what sort of contaminants of emerging concern are, but again, just to provide a bit of an overview of why it's important to sort of understand what these contaminants are and their risks, is because again, there's currently just not enough literature and information out there to provide, I suppose, a thorough assessment of the risks that these different new emerging or current contaminants have. So emerging contaminants, they're detected in the environment at concentrations that are significantly higher than what you would expect, and they may cause either ecological or human health impacts depending on the organisms that are exposed to them. Their risks to both humans and the environment is not fully understood, and as I mentioned, this is where there's just not enough scientific literature and understanding out there, and so we have this high level of uncertainty surrounding sort of their presence and risks when we think about them out in the environment. And then the OECD has actually included an updated list that includes industrial and household chemicals. Brad will be talking about some of these examples shortly. Ben's already touched on the PCPs, or pharmaceutical and personal care products, but this class also includes microplastics and nanoplastics, different agrichemicals, as well as nanomaterials, and then another class of other that sort of falls outside of those definitions and that umbrella. Another type of contaminant that has constantly caused increased risk to aquatic environments as well as human health historically is heavy metals, and heavy metals are still a concern, and we do detect heavy metals at high and elevated levels in a lot of developing countries where again there are these increased risks from toxicity, and some examples of this include in Southeast Asia where high cadmium levels that are present in both water and food sources and contamination are actually causing increased incidences of chronic kidney disease in the human population. So we know that metals do have these high risks to both environmental and human health. And so it's important to still monitor and predict and look at the risks that these heavy metals pose. With heavy metals, we use them in a variety of applications. They're used so widespread that there are varieties of different sources that can introduce these metals into our aquatic environments, from terrestrial runoff, as well as from direct inputs as well, from either wastewater treatment plants, from urbanisation and urban landscapes, from industrial applications, stormwater runoff, from mining applications as well as from agriculture. And especially in agriculture, a lot of metals like copper, zinc, and some other metals that are micronutrients can be used in fertilizers. But we also know that if you go out and if you've got a veggie patch at home, if you look at some of the ingredients in some of the insecticides as well that you use in a veggie patch, there's usually either copper used in those products as well because they are toxic to different insects and pests. And so again, we do know that metals are still used in a variety of different agrochemicals, either fertilizers or insecticides. And so there is this potential for these different metals to be introduced into the environment. But more importantly, we also have historical legacy sources as well, with recreational activities as well as other past industrial sources and applications as well. When we think about heavy metals, though, it is very complex. So the risks of metals is very much dependent on water chemistry. And so generally, heavy metals become more toxic to aquatic animals when the water is soft, in more acidic environments as well. And so there are a lot of chemical factors that can play an important role in dictating the actual toxicity risks of these metals to the different environments and the organisms that live there. So with Lake Colac, we know that Lake Colac has important significance to the local and surrounding communities for recreational activities, but more importantly, it's also home to a variety of nationally threatened and Indigenous plant species and animals, where it's home to the vulnerable growling grass frog, as well as the Corangamite water skink and the critically endangered curlew sandpiper. So it not only has a lot of actual importance for the local community for their recreational activities, but also it's an important actual freshwater catchment and site that has ecological significance as well. Lake Colac's also an actual unique freshwater source because it actually receives water inputs from different sources, from the underwater groundwater and aquifer, from rainfall that again can flow from sort of the different agricultural areas into the actual lake. You also get supplies from the town's stormwater and then discharges as well from the wastewater treatment plant. And so because we have this complex environment, there is a variety of different opportunities for contaminants to enter this lake via these different sources. And so that's why we've chosen this site to actually study it, to try and predict and look at whether there are any environmental contaminants present and then where they may be coming from. Excuse me for a second, I'm just getting a cold, so Hopefully that's helped. So Lake Colac, as you can see in this satellite image, it's sits right adjacent to sort of Colac, and you've got the Colac foreshore here. Now, the actual lake itself is surrounded by this vast sort of area of agricultural as well as dairy sort of sites, and so there is a potential for a lot of nutrient as well as other runoff coming from these environments here. It's supplied by two creeks as well, the Barangaruk Creek and Deans Creek that we can see here. And more importantly, another unique thing with this site is it's not a positive, unique sort of example or situation, but there used to be an actual tip or dump located just about here, sort of near the actual boat ramp. And so that's now been sealed over, and it's been sealed over for a number of years. But again, we don't know what was actually buried in that dump or site and whether anything's leaching from that as well. And that could be another source of contaminants as well to this freshwater environment. When we think about Lake Colac, as I mentioned, there's a variety of different ways that contaminants could enter this actual system, from agricultural runoff, and this can include pesticides, insecticides, as well as nutrients, heavy metals. We have the town stormwater that can also put inputs of different contaminants as well. And then finally, wastewater treatment plant outflow that could also introduce personal care products as well as microplastics, nanoplastics, or industrial chemicals. So this is why we've chosen this site for our study, because again, it is quite complex and there are multiple potential sources of contaminants that could be entering this actual environment. So the overall project objective, so the first part of the project is trying to actually identify and then quantify the concentrations of different contaminants at different locations in the lake, but also across a seasonal time point. Within this project, we're measuring heavy metal concentrations. We're also looking at microplastics and pesticide or agrochemical levels. Ben and Sam are working on the personal care products and pharmaceutical component. And then overall, by measuring these contaminants in these different sites, what we're hoping to do is try and identify and predict where these potential contaminants are coming from. And that information is going to be really important so that the local community councils, as well as Barwon Water, can then use that information to implement future strategies to try and minimise the inputs of these contaminants to the lake. Another objective that we're looking at developing is what's called a bioeffect index. Now, what this will basically do is we're using freshwater shrimp as the model system in the lake. And so those freshwater shrimp, we're looking at a variety of health indicators. So from the cellular, molecular, up to sort of the physiological as well as whole animal health status of those shrimp. And by looking at all of these different sort of parameters of the fish, of the shrimp's health, we're going to develop what's called this effect assessment index to try and then provide an overview of the actual health of this local ecosystem. And this bioeffect assessment index is something that we're hoping to then roll out across other catchments in the future as well, when we can sort of look at measuring similar things in different aquatic organisms as well. And then finally, another part of Sam's project that Ben touched on is looking at the trophic transport of pharmaceuticals and personal care products. But for today, I will be just focusing on some of the results that we now have in the heavy metal concentrations. So out at Lake Colac, we've selected 8 different sampling locations. So on the right-hand side here, you can see each of these. We've got the first site is at the Lake Colac boat ramp. We've got the Stoddart Street Park, where it's directly next to a stormwater input and a stormwater drain. We have Rosemoyne Park and then Meredith Park, which are sort of more of our north and northwest sort of locations. We sample in the centre of the lake as well. And then we've got a location near the wastewater treatment plant outflow, as well as the 2 creeks that are bringing water into the lake with Barangaruk Creek and Deans Creek. At each of these sites, we've collected surface water samples in triplicate, and then at all of the sites just within the actual lake environment, excluding the central part of the lake, we've also collected flora and fauna. So the flora component is what Sam and Ben at Fed Uni will be working on, and then we've collected shrimp or freshwater shrimp samples, which is a species that we think is Paradia australiensis. We're doing some genetic testing to find out whether it is this specific species, to find out what the freshwater shrimp population is like there. But we do predict that it's this species here, and it's a really good bioindicator model for looking at sort of contaminant levels and health as well. And so we've collected sort of the flora and fauna, or the shrimp species, from each of these sites here. We've done a seasonal assessment. So we went out to the lake and collected samples over a 12-month period that started in mid 2022 and ended sort of early last year. And we've now been, or our students have been, working hard to analyse all of these samples in the lab. So the way that we actually process the samples for heavy metal analysis, with the surface water, we're able to just filter them to remove any larger organics and other suspended sediments. This doesn't remove it doesn't remove a lot of the actual metals, and so we use a 0.45 micron filter. We then acidify the samples with nitric acid, and we analyze them using independently coupled plasma mass spectroscopy, which is this instrument here. And this is able to do full trace metal screening, where we have an actual standard and library where we can screen a whole list of elements and metals in one go. And so it's a really robust method. We also incorporate National Measuring Institute reference standards, so a prawn standard as well as a water sample reference standard, to ensure that we've got our quality control checks in place. For the freshwater shrimp, this is a little bit different in the process. We actually dry the shrimp in an oven for 24 hours at 70 degrees. We then get the dry weight of the shrimp. We digest them in a mixture of nitric acid and hydrogen peroxide at elevated temperature for 4 hours. This fully dissolves and enables the actual organic components of the shrimp to actually be fully digested. We're then able to then analyze those samples as well on this instrument to be able to detect metal levels and element levels at very trace concentration, so down to parts per billion levels, so really low. We've also been conducting water chemistry analysis out at the lake. And as I mentioned, this is really important when we think about the effects of metals, because we know that different chemistry components of the water can dictate the actual levels of metals that are accumulated and taken up by animals, but also their toxicity risk to aquatic organisms as well. And I'll talk more about this in a bit more detail later in today's lecture and seminar, and the significance of that. When we look at a bit of a snapshot, so this is just some heavy metal measurements in surface water levels from sample time 1. And you can see that for a lot of high risk metals, so our aluminium, arsenic, cadmium, nickel, lead, and silver, they're really low to even below detectable limits. So this is actually really good news, because a lot of these metals are really toxic to aquatic organisms, but more importantly, most of these metals are toxic to humans. And so these are the high risk ones. And this is really positive news, that we've got really low levels and even below detectable limits of some of these really nasty heavy metals present in the surface water. We've also measured these in the shrimp samples, and they were very low and below detection limits for most of these metals as well. What was interesting though is we did see elevated levels of copper. So on this graph here, we've got the copper concentration in micrograms per litre, and we've got on the x-axis here, or down the bottom, is the different sample sites. The different coloured bars are our different sample times, so sample time 1 through to sample time 4. And so in our surface water samples, you can see that we were getting concentrations ranging from 1 to 25 micrograms per litre. Now, when we think about these concentrations, it's important to think about sort of the trigger values as well as the potential toxicity concentrations that we see in different species. And so when we take these values into consideration, the trigger value here in the blue line, this is the concentration at which anything beyond that could be potentially causing toxicity risk to those local organisms. And then if we look at the LC50, so this is the concentration in crustaceans at which 50% of the animals would actually have mortality or die, you can see that these concentrations that we're measuring are well above the trigger value as well as the LC50 value. And I'll touch on this in a bit more detail and why sort of these values are important, but more so as well when we look at the water chemistry, there could also be some protective effects happening as well when we think about the animals that are living there. One thing that I will point out, sorry, I know I'm jumping ahead. So another metal that, we also measured copper levels in the shrimp species. And so in the shrimp, you can see again that there were elevated copper levels. We've measured these as milligram per kilogram, so the mass of the actual shrimp. And just to put this into perspective, the actual concentrations of copper in our NMI prawn reference material, these concentrations here are about 5-fold higher than what you would expect in a sort of healthy environment and a shrimp species in a healthy environment. So they are quite high compared to what you'd normally see. If I draw your attention to this region here, though, an interesting result that we saw, and this was for both copper and the zinc that I'll mention about shortly, what we actually saw is it appears as though the creeks that are feeding into the actual Lake Colac, so Barangaruk and Deans Creek, they had elevated levels of copper at sample time 1. And it appears as though when we've done some other statistical analysis, I won't be presenting it today, but through some other statistical analysis we've done, it's looking as though the actual creek could be a point source for these metals coming into the lake. More importantly, after sample time 1, we had record rainfall in the area over that 12-month period. And so what's actually happened is we've introduced the copper into that lake after sample time 1, and you can see that it actually distributes quite evenly across the lake, and we weren't seeing any differences from any specific locations, which was really interesting as well. We did also see elevated zinc.
Aaron, we're probably going to have to skip the zinc results. We're running out of time. You've got 1 minute.
Perfect. So with the hazard quotient, Ben presented on this as well, we did see elevated levels. So anything above 1 is a potential risk. And so for both copper and zinc, we did see elevated levels. And then more importantly, what's important is we've actually seen when we've been out there sampling, it is still a healthy environment. So we're getting large populations of shrimp that we're able to catch. And we are looking at health indicators of the shrimp, and we'll have that research and results to share in the future as well. But what I want to show is that Lake Colac itself is a little bit alkaline, and it is extremely hard. So although we have elevated copper and zinc, it is highly likely that that copper and zinc is actually being is binding to these other organics and materials and elements in the water, and that the hardness is probably protecting the animals that are present within there, which is interesting in itself as well. So from the project findings, we've shown that a lot of the high-risk metals are in low levels, which is great news. There were elevated levels of copper and zinc. However, it looks as though there is this protective buffering occurring in the lake from the hardness and the pH. And more importantly, it looks as though we're getting these metals, the copper and zinc, potentially coming from agricultural runoff or stormwater that's coming into those creek systems and leading into it. So with the future research outcomes, all of the information that we're collecting and will continue to collect over the next 12 months will, help inform decisions from the local council, CMA, Barwon Water, and local communities so that we can try and better protect the environment by minimising these contaminants entering the actual lake, but also develop strategies for remediation that Ben alluded to and spoke about as well. And just a bit of a shout out as well. My group, again, we've got projects occurring across multidisciplinary areas across this One Health approach, looking at human health risks and environmental and animal health risks of different contaminants as well. So this is just a shout out to all of my amazing students that are doing a lot of work in this space, and without them, I wouldn't be presenting the data today. So acknowledgements as well, everyone that's involved in this project, as well as Nishat. All the data that I've presented is her data. So thank you, Nishat, for sharing it. And thank you as well, Ben and Barwon Water. It's been a great project, and we look forward to releasing and sharing more results in the future.
Well, thanks very much, Will, and thanks, Aaron, for this very important work that you and Ben are doing. It's important that we track pollutants down to their source, and hopefully we'll be able to stop that pollution occurring. To complete today's presentation, Dr. Bradley Clark of Melbourne University is an Australian authority on emerging contaminants in aquatic environments, including PFAS and microplastics. Dr. Clark.
Great. Hopefully you can hear me, you can see my presentation. It's great to be here today. Thank you very much for the invitation, friends of Barwon, Lachlan, Will, and Barwon Water. It's my absolute pleasure to be here. It's been great to hear some of the work by Ben and Aaron. I actually grew up in Ballarat and I spent a lot of time in Barwon Heads, so I sort of know the area reasonably well. I'm going to talk to you for 20 minutes. I'm going to go big picture and talk about PFAS and then microplastics and then come back to the big picture. So I'm going to start with a quote. For the first time in the history of the world, every human being is now subjected to contact with dangerous chemicals from the moment of conception until death. I'm going to tell you who said that quote at the end of this talk. Pollution is the largest environmental cause of disease and premature death in the world today. Pollution causes 9 million deaths worldwide each year, or 1 in 6. And pollution is responsible for more deaths than AIDS, tuberculosis, and malaria combined, and 15 times more than from wars and other forms of violence. This paper was pre the wars that we're seeing in Ukraine and the Middle East. But I also want to draw attention to a problem, and it really is related to chemistry. So what we have, chemistry is really the foundation science of modern life. And so all of the innovations that we really enjoy are due to humanity's ability to manipulate matter. Now, this is a really fantastic thing, but what we're seeing is actually that the amount of chemicals is really being introduced is really sort of spiraling out of control. So the chemical industry is predicted to expand rapidly by 2030. And one of the big and key issues here is that we used to think there's about 100,000 chemicals in modern society. Actually, that number's closer to 350,000. And over 100,000, we don't know what they are. And we don't know what the effects are of many of these chemicals. So this is a real significant and emerging problem. And why? And some of the worst of these chemicals, they accumulate in our bodies and they can cause things like cancer, reproductive health problems, impaired immune systems, and neurological damage. You may have seen in the paper just last week about pregnant mothers with high levels of bisphenol A, which is a plasticizer, is related to autism in boys that were born. So what we also see is this intergenerational effect. This study that I'm showing you on the screen is where we have side-by-side villages in Mexico, and what they're showing is exposure to pesticides actually has cognitive damage as well. So it really is this risk that we're playing and a gamble that we're playing with our own health and the health of future generations. PFAS is a really great case study for understanding this issue. So PFAS are organic fluorine compounds. You've had them in your house. The things that you probably have are 3M products, Gore-Tex. You might have a non-stick fry pan, and it's Teflon. You probably used to spray your shoes and your couch with Scotchgard. You've probably got a can of it in your laundry. It was born out of the Manhattan Project, so where humanity created the atomic bomb. From there, we had a non-stick surface that then got incorporated into the frying pan, was where it really began, and it got incorporated in a bunch of different products in our homes. In actual fact, 3M and DuPont became aware that it was toxic in the 1970s, but concealed that fact for another 3 to 4 decades. In 2001, we discovered the omnipresence of PFAS, one of these chemicals, throughout most humans and animals and almost all environmental media. This led to phase outs of particular types of PFAS, which I'm going to talk about as well. And so we used to have about 2, and then all of a sudden we started to get replacement PFAS being introduced. So now we think that there's somewhere around 9,000 to 15,000 different types of PFAS. And really it's probably one of the most significant environmental issues that Australia is currently dealing with. And really what's happening is the cost of the issue is being displaced to the taxpayer. Now, I know that people probably won't have chemistry, but so this is a carbon fluorine bond. It doesn't exist naturally in nature, and it's virtually indestructible. But on the right-hand side, if you can see, on the right-hand side, this part here will break down. And so our innovative chemists have made a product that has chemical that will break down in the environment and transform into the things that we're concerned about. So these are the first two. You've probably seen these in the paper. This is what they're talking about. It's called PFAS. This is what it looks like. So it's like a fatty acid in a way. It's got a carbon backbone where all the hydrogens have been replaced with fluorine, and then it's got a functional group. Now, we know now that it causes a range of health impacts, including PFAS is being classed as an International Agency for Research on Cancer as a known human carcinogen, and PFAS is a suspected human carcinogen related to testicular cancer and kidney cancer. And there's a range of other things that it causes, including immune impaired response. It's also associated with lower birth weight for children, and typically lower birth weight is associated with long-term health impacts. So these chemicals accumulate in our body. So if I took a sample of anyone watching, you would have this circulating in your blood. It's mobile in the environment, and it's persistent. We don't really know how long they'll last for because they're not naturally occurring in any way. Now, again, this is just a slide for some of the chemists. Maybe you go back to your high school chemistry. What we're seeing is this proliferation of novel substances that are being introduced. Almost all we know nothing about. And this is a really concerning situation. This one here, hopefully you can see my laser pointer. Now this one here is, so we've got a mixed fluorinated, we've got ether, and we've got this chlorine. This one is, we found in my blood, and I'm the first person in Australia to have this reported in their blood, and it's a PFAS replacement. And I'm going to draw your attention to this novel one up here too, which I'm going to talk about in one of my studies. In Australia, you've probably seen this, the main issues have been associated with the Defence Forces. The Defence Force had been setting jet fuel on fire with AFFF and then putting it out with AFFF. And that resulted in contamination of groundwater in adjacent communities to these areas. PFAS is everywhere, and this is one of the big issues. We're going to be dealing with this for decades. So industry and Defence Force and airports were the primary emission sources. But the other sources are actually your home. It's all through your home. And then wastewater treatment plants become now an emission source or a point source to the environment. And the other one is landfill. In actual fact, there's often a link between landfill and wastewater treatment plants because the water that circulates through the landfill is often sent to wastewater treatment plants with time. So I'm going to show you some data. There's lots of acronyms. Don't worry. You can just get the idea. So this was a study where we looked at PFAS, so a bunch of different PFASs in treatment plants across Australia. We had 19 different treatment plants from urban and rural and all the different states. And if I'm going to draw your attention to the mean concentration, so we took it at the start of the treatment process, it had 90 nanogram per litre, and at the end of the treatment process, it had 140. This is our mean concentrations. And you can see the range as well also increased. And we thought that was very interesting. And it's almost like the PFAS is being formed through the treatment process. And that's exactly what was going on and what we observed. So these are various chain lengths. And what we're seeing is that they were actually being formed by those precursors that I pointed out before. And so we are getting novel substances that we don't know what they are, degrading through the treatment process into the ones that we're concerned about and we are regulating. This was a pretty cool little study we did as well, where we wanted to understand the source of PFAS. So what we did was we took a sample every hour at the influent of a wastewater treatment plant. Now, what we have here is a plot of days of the week, Monday to Sunday, and you can actually see that the profile is different. Now, on this right-hand side, this is really important here too. So what we see is our baseline level of PFAS. This is the main one we regulate in Australia. So this is the amount that's coming from our homes. And then all of a sudden, you can start to see some spikes. Our first spike event is Friday afternoon. Our second spike event is Saturday on Saturday morning. This suggests to us that this is being used for some sort of timed event, possibly industry, And it's really important because this means that we can actually control the source. If we can go up through the sewer and not have it discharged in the first place, then we won't be releasing it into the environment afterwards. Biosolids are a really interesting situation across the country. About 70% of the residual sludge from wastewater treatment plants is applied to agricultural soil, and then it can be laden with pollutants. And this was a little study of 19 different treatment plants on PFAS as well. And it's a little bit hard to explain with analytical chemistry, but analytical chemistry can only see what we've sort of built a method to see. And so if you don't have the compound in your method, you don't know that it's not present. So you have to build a method, you have to buy a standard, build a method, and do your quantitative work. And we'd added this thing called a DIPAP into our method. Now, DIPAPs are actually used with paper. Now, these are one of these ones that are probably going to accumulate more because takeaway food packaging often has to be oil and water repellent. Now, so you think about your pizza boxes. Now, so it's get lined with particular things, and it could be lined with a DIPAP. Now, what I'm showing you here is we have a fully fluorinated section, and then we have a bit in the middle that looks more organic in structure, and it will break down. And it breaks down in the environment to things that we're worried about. So this is something we should be paying attention to moving forward. And another study I'm going to show you is a little study we did at Albert Park Lake. We were interested in the relationship between the levels in the water and the body burden in the swans actually on there. And we found that Albert Park Lake was surprisingly contaminated. We still don't have a good explanation for that. But one of the things that was really strange was we just added this new compound. That was that cyclic version I showed you earlier. And we found that that was present in all of the sites that we studied. And in fact, it was present in the birds. Now, the really good thing about this study was that we actually analyzed their feces as well as their blood. We actually showed that there's a clear relationship of the blood and the feces, which means that you can sample the feces of the birds, just probably opportunistically collecting them. And this would give you a good estimate of the internal dose. So the question really is the Grand Prix the source? So there's only one known use of this chemical, and that is as a hydraulic fluid in the airports. Now, Albert Park Lake is nowhere near the airports, so we're trying to figure out where this has come from. So we've done a follow-up study, and in actual fact, we're still no closer to determining if the Grand Prix is the source of this. So we're still trying to figure this out. Now I'm going to talk about microplastics in my remaining time. Microplastics have captured the public's attention like nothing else. And in fact, you'll see this in the news almost every second week. And partly is now because it's being found all through our bodies. We used to think that basically it wasn't biologically implausible for plastic to be accumulating in our body. But actually what we've seen recently is it's been found in liver, brain, heart, blood, testicles, and seminal fluid. So we're seeing this distribution of this plastic residue all through our bodies, but also we're seeing this all through the environment. So a lot of what we produce on land will end up in the ocean. We see cases of entanglement with animals, and in fact, this is being proposed as a geological indicator of the Anthropocene era. And what we really need to understand are the sources, exposure, and impact of microplastics on human health and the environment. And the reason it captures the public's attention are because of images like this. I'm going to draw your attention to the one in the middle, and this is a shearwater. It's a little baby, and what is happening is that its parents have gone foraging the ocean for food, and then they bring that food and they've eaten this plastic thinking it's food. They're taking it back to the nest. They've regurgitated it, given it to the bird. And the babies don't have the ability to regurgitate this plastic once it's in their system. And then you see these horrible images of animals that have starved to death with plastic in their stomachs. Now, even as we start to think about recycling, it's really just a temporary measure. So we need to be thinking, we do need to be thinking about strongly changing our behavior related to plastics. Now, one of the things that's likely to happen as we move away from fossil fuels, and this will be really perverse, it is that the fossil fuel producers will try to push that material into polymer production. In fact, they'll also try to push that into chemical production as well. And so we have to be vigilant and mindful of these as potential issues. Plastics are really complicated too. They're not just a single chemical. There actually can be things that absorb other things. Like, so in the environment, polyethylene is actually like a magnet for other chemicals. And that's really concerning. So they don't fit into our traditional models of how we understand risk, like Ben showed with his risk quotient. I'm going to show you the results of 2 studies in my remaining time. The first one, we were actually asked by a local community in Bacchus Marsh to measure the concentration of PFAS in bores in their town. They were worried about the Westgate tunnel spoil being put into the local landfill. We didn't find an issue with PFAS, but actual fact, we measured for microplastics, and we were the first group in the world to find microplastics in groundwater. Once we published this has now become very well documented that microplastics have now found their way into groundwater. And that to me is a really sad indictment of humanity too, and of how badly we're contaminating the environment, because there's no way really to remediate these groundwater bores. And what we also see from bore 3 is we can actually link the land use, the surface land use, to what we're finding in the bore. And the last study I would like to share with you is, it's a bit of an old study that I did, but I think it's really a good one, is that, as I say, plastics in the environment will act as a vector for other chemicals. So that they're like a magnet. They'll concentrate them by a million or more, because a lot of industrial chemicals don't really like being in water, and particularly the ones that accumulate in our body. And so we ran this experiment where we fed fish microbeads that we'd taken out of a cosmetic product, and we dosed it with a typical level of contaminant that's been reported globally at this 200 nanogram per gram. And we fed fish, we put this into their food, and we fed it to them for 60 days. And what we showed very clearly is that the plastics do act as a vector for the transfer of the chemicals into the tissue of the fish. So not only are the plastics themselves an issue, we also see that they are a vector for other contaminants. And unfortunately, when we combine this with wastewater treatment plant, all of the chemicals we use in society go through wastewater treatment plants and landfill. And so we've got this real opportunity for the concentration of novel and unknown substances. Now, I'm going to end with, I started on the big picture. I'm going to go back to the big picture. We are gambling with our health, and it's getting worse. The situation is getting more, it's getting more severe. Now, if we can't make the argument that we need to keep the environment clean for the sake of keeping the environment clean, we might be able to make an economic argument around this. And it's estimated that the welfare costs associated with pollution are in the order of $4.6 trillion per year, which is 6.2% of global economic output. When we look at things, the health effects of just a small group of chemicals in the USA where there's consensus agreement on the health impacts, it's thought to be $340 billion per year. And if you translate that number to Australia, we're talking about 30 or 40 billion as well. And so there's substantial economic costs associated with pollution, amounting to a small percentage of the overall GDP. And this means that we should, if we use that money in a preventative way, we can actually be saving money. And also the cost of the human cost of some of these things, say the autism in boys. Okay, that quote I showed you at the start of my talk was actually from Rachel Carson's The Silent Spring in 1962. We're talking about 60 years later, and it's actually, things can't be more true, but it's actually, there's more chemicals that we're in contact with from the moment of conception until death. That number, 275 million, is the number of unique substances registered in the CAS database and that are known to humanity. I want to thank my team. They're the people that did all the work. And with that, I am done. And I think I hit 20 minutes exactly, Will.
Well done, Brad. Great work.
Thanks, Will, for moderating today, and many thanks to Aaron and to Ben and to Bradley for a wonderful and somewhat scary presentation. Friends of the Barwon, really hope that you enjoyed this webinar. Please join us for Pollution webinar number 2, the flyer for which will be emailed in the next few days. Topics for this webinar include pesticides in our waterways, more on discharges from wastewater treatment plants, and will feature a current example of pollution dilution, which asks the question, is the concept of dilution ratio simply a license to pollute? As you leave today's webinar, there will be a short survey. I hope you can take a minute to complete, and we look forward to seeing you next time.